clearly there was a need for this type of symposium, just looking at the whole room full of excited people. So it's really fun to be here. I don't have, do I have a, oh, this, yeah. Okay, so I think for everybody in the room it's clear that the infection by an RNA virus is really a process of evolution. It's evolution at work. So the virus that you get infected with is different than the virus that navigate from tissue to tissue that eventually reach the tissue where it causes disease. And also is different than the virus that is shed into the environment to infect the new host. So, and it's clear that evolution is driven by mutations. But one of the things that is sometimes not obvious is that a number of experimental data suggest that most of the mutations that arrive randomly will be detrimental, will reduce the fitness of the virus. So the virus needs the mutations. On the other hand, it has to tolerate the mutations. And it's this very subtle balance between producing mutations and tolerating mutations where the trick for an effective evolution, viral evolution, stands. And so now we know that virus populations are not constituted by a single genotype, but it's a collection of mutants. And as the virus replicates and is passage from infected cell to infected cell, a number of mutations arrive by this error doing replication, and some of these mutations will be eliminated, and at the end you will end up with a collection of tolerated mutations that constitute this diverse population that sometimes is called quasi-species. When you sequence your RT-PCR in a virus population and you sequence, what you end up having is a consensus sequence that represents clearly the virus that you're looking at, but it ignores completely this enormous diversity, genetic diversity that is in the virus population. And it's not only about a swamp of mutations, but the critical concept in the quasi-species theory is that these mutations that are in the populations, they have genetic interactions with each other. So a perhaps better representation of the structure, the genetic structure of the population is given by this mutation network. So, at the beginning of the replication, you start having these 3 mutants that are separated by 1 or 2 mutations away, and they start to form these networks that gain in complexity as the virus replicates. And the important thing to remember here is that each mutant is related to the next one by these connections. And sometimes to get from this mutant to this mutant, you have to navigate through other mutations. And the important thing is some of these mutations might actually reduce the fitness of the virus, and it may be completely unacceptable to acquire these mutations. So maybe the connection between this mutant here and this mutant here has only one evolutionary trajectory possible. And so it's about how the virus explores sequence space that allows the virus to acquire this diversity and have an effective adaptation to these dynamic environments. We some time ago showed that in our lab, in Carla Kierkegaard's lab, we identified mutations that have high fidelity of replication, so polymerases that don't introduce so many mutations per replication. And so these networks were restricted as opposed to compare with the wild-type network. And what we show is that when you have a this population that is not very diverse, the virus, the virus population was not pathogenic. And you needed a much more diverse population to cause disease in a mouse model. So clearly, diversity is very important for this virus to access to the organ in which cause disease. The virus that we mostly work with is poliovirus, and it's a neurotropic virus. To the target organ is the central nervous system. And the mouse end up dying either with paralysis or other neurological effects. So the first question I wanted to ask is, or present to you is, can poliovirus, this RNA virus, accept this large amount of mutations that theoretically are generated of each round replication? So how is it possible that the population looks so diverse? And so we developed a sequencing method in collaboration with Joe DeRisi, and Dave Webster and Armin Heckley in the lab put together this approach. This was published a couple of years ago, and it's totally obsolete. Nobody would use it anymore. And this is the pace that this technologies go. But this slide actually shows something interesting. So what we are doing here is taking poliovirus and pass the virus in mutagen, this nucleoside analog called ribavirin. And we do passage 1, passage 2 in no ribavirin, 100 micromolar, 400 micromolar, and 1,000 micromolar. And here we sequence the populations, and what it represents here is mutations that appear during these passages. And what you see is that as you grow the virus in higher mutagen, you accumulate a large number of mutations. And so these are viable viruses that really can tolerate a large mutation load. Okay. So what is the phenotype of that? So Adam Loring in the lab developed this assay to try to, similar what Eva was discussing today, how can we correlate the genetic diversity with the fitness of the virus? And so Adam borrowed from yeast genetics a technique, which essentially is you take different genomes and barcode it using oligonucleotides. And then you can follow this genetic, each of these genomes using a microarray setting. And so essentially what the experiment is, you take a poliovirus population, grow them in mutagens, so you increase the diversity, so you will be somewhere in the middle of this high load of mutations. And now you take several of these viruses and insert it two barcodes, two oligonucleotides, so you can follow this. And in general, sequencing analysis demonstrate that you have increased about 4-fold the number of mutations per genome. So this is a heavily mutagenized population. And the question here is, what is the phenotype of these populations? Now you increase the mutations. Can the virus maintain a good fitness compared with the wild type? And so what you do now is mix this population with wild type and pass the virus in tissue culture. And essentially, you can obtain this kind of graphics with high precision, high quantitative precision. So you look at the frequency of each individual. For example, this purple virus here you can follow because they barcoded over passages. And so this is doing pretty well. The blue is doing less well. And then there is a couple that seems to be losing in this theoretical curves, not doing so well with respect to the other guys. So when you graphic all this virus population, the fitness of this virus population, what you see is that in general, you see about the average of these viruses, the fitness, is about 60 to 80% wild type phenotype. So they can the phenotype of this fitness value is about 0.6 to 0.8 of the value 1 corresponding to the reference wild type. So this argues that not only the virus accept a lot of mutations, but many of these mutants that are being produced during replication do not have a very strong phenotype due to the mutations. And so this brings the point that I actually wanted to discuss today. And this is about this mutational robustness. For a virus to be able to evolve, not only has to generate mutations, has to tolerate the mutations. And so what could be the mechanisms for this RNA virus robustness? One possibility is just the proteins that viruses carry are robust, are designed in a way that can tolerate a lot of mutations. The structure, the core of the protein is done in some way that the structure is robust enough and can tolerate a lot of mutations. In RNA viruses, we know that we always operate in a very high number of individuals in the population. So you have 10 to the 9th platform units. And so this allows the virus for a number of possibilities. One possibility is that the virus doesn't care that 90% of their progeny, their kids, are dead. So if only 10% alive, that's OK. The other possibilities is because they actually operate at this high large population levels, they can have this kind of genetic gymnastics, like a complementation and recombination. So in other words, you can have two viruses that are defective, co-infected in one cell, and they share proteins. And so in this genetic complementation, two viruses that are not doing very well can actually produce very good progeny and replicate very well. Or they can recombine their genome, purging their mutations in this way. But there is this other concept that we don't think about it very often, is that the virus might actually exist in a place and sequence space that allows for the introduction of mutations without much change in the phenotype. So virus might live in an area and sequence space where there is a high density of neutral mutations. So in a graphic form, what I'm saying is that here you have two peaks, fitness peaks. So this is fitness, this is sequence space, and this is a very simplified idea. And this was put forward using mathematical simulations by a number of people, particularly Klaus Wilke. And in these two peaks, here you have a peak that has very good fitness. So all these viruses are replicating very well, even better than these viruses in here. But as they start introducing mutations, these guys lose fitness very quickly. So in few mutations, you are in the bottom of the fitness peak, whereas this guy seems to be able to accumulate mutations and still maintain a certain degree of fitness. So this theory was called the survivor of the flattest, because the flattest your curve is, the more mutations you can tolerate. Now, there's a problem here. And the problem is that if you introduce mutations but your phenotype doesn't change, what good does it do to your adaptation? So, okay, this is the old joke when the housing market was good. Now it's kind of sad. But anyway. Anyway, so going back to this mutation robustness. So if your robustness, if you can introduce mutations but it doesn't change your mutations, seems to be opposite to evolvability. Seems to be that you can introduce all mutations that you want, but you're not going to evolve much. But Jeremy Draghi in Josh Plotkin's lab in UPenn has put forward this concept that I think articulated mathematically this concept that I think it sheds some light of how living in a neutral space can help you to evolve faster. And the idea is that you have your network of mutants, and if you just focus in one area here, you might have two mutants here that are neutral. In other words, this mutant here and this mutant there doesn't change your phenotypes. They are silence mutations, okay? So there's no change in your fitness. But if you're here, you access to these green, blue, and gray mutants that might give you an advantage. So this is an advantage by the neighborhood in which you live. So for example, if you have arginine codons, these are two possible codons, but the number of one mutation away mutants that you can generate with this codon is very different than the type of mutations that you can generate with this other codon. So this codon allows you this, allow access to this neighborhood, whereas this allows it to this neighborhood. So Adam decided to try to experimentally test this concept. Is it possible? Is it true that when the place where you live in sequence space allows you to evolve better or worse? And so the idea is to take the virus that is living in one place in sequence space and move it to a different place in sequence space. And so Adam borrowed viruses that were the controls of another experiment that Eckhard Wimmer did a couple of years ago. And that was these two viruses, MAX and SD. So this is poliovirus, and these mutations were introduced in the wild type, in the capsid region. So here you have a number of codons that encode for the capsid proteins. And these two mutants, what it is a swamp of codons. So it's introduction of a number of silence mutations in this region without changing of amino acid sequence. Okay? For example, SD is simply taking one codon that was encoded here, leucine, and moving it down here, and taking this codon here and put it up here. It doesn't change at all the codon, the GC contain. The only thing it does is change the position where the codon is. Okay? This other one here is using optimized codons. So it's just simply using an algorithm, and you say in human cells, these codons are preferentially used, so we're going to change some of these codons for optimized codons. So this gives you max. Now, the important thing is now you're moving your virus population 900 positions away from the original position of the wild-type virus. There is no functional RNA elements in the poliovirus capsid region. You can delete the capsid regions, and the virus replicates just as well. The important thing is that when you look at the codon usage, and these are different codons for wild-type MAX and SD using this color code, and you can see that there is no much differences. It's not like before you were using a different codon, and now you are completely changing the type of codons that you use. Okay. Then we characterize these viruses in tissue culture, and the first thing we did is to look at protein synthesis. Since this thing is encoding for capsid proteins, we wanted to know whether these viruses actually can produce capsid proteins. And you see here by an S35 protein gel, and this is the quantification. There is no difference between wild type, MAX, and SD for any of the capsid proteins. In terms of RNA synthesis, this is an RT-PCR, quantitative PCR assay measuring a number of copies of RNA genomes, and you see there's no difference over the course of infection in HeLa cells. But there was a difference, and the difference was in the fitness distribution in a plaque assay. So essentially, everybody has done eventually sometime a plaque assay. So you infect a monolayer of cells and you see different plaques. And there is a little distribution. There's some plaques that are larger than others. And so what Adam did was develop an approach in which you take a picture of the plaque and then measure the size of the plaques. And that way you can actually analyze many, many plaques and measure many, many plaques. So here we are comparing wild type in purple and max in yellow. And what you see is that the peak of the, this is, I think, probably 36 hours post-infection at 37 degrees. So between 3 and 3.5 pixels, which is not inches or centimeters, is pixels. So it's a new measurement. But anyway, the point is that this is wild type here, and max is a little bit larger plaques, but not significant. But SD is very is smaller. You see this SD has a smaller plaques between 2.5 and 3, whereas wild type was here. And the other thing is interesting is that the distribution of SD is much larger. So it has many more smaller plaques than larger plaques. So it seems to be that when you infect cells with single genomes, you have different outcomes of infection. In other words, in a plaque assay, you dilute your virus sufficiently that each cell will acquire one genome. And in this assay, what it's suggesting is that the mutations that are being accumulated during replication, because as SD is accumulating mutations as it replicates, it will produce a much larger distribution of phenotypes in general, making the plaques a little smaller. So this might suggest that SD is less tolerant to mutations. But the experiment that actually demonstrates this is if you take these viruses and put it in the presence of this mutagen, and you measure how resistant the virus is to the mutagen over a course of different concentrations of the mutagen. And what you see here is that here is max and wild type, and SD is indeed less tolerant to the mutagen. So it's less robust when you increase the amount of mutations. This line here is a control of one virus that is high fidelity and has higher tolerance. Okay, so what we're suggesting is that the SD has moved 900 nucleotides away from the original wild type position in sequence space, and now has a much more sharp peak and is less tolerance to mutations. So we wanted to document this, and so we started to examine this using deep sequencing approaches. And so the setup of the experiment is you take wild type max and SD, and you pass it at between 0.1 and 1 MOIs, 0.1 to 1, and then you sequence the populations after 3 passages. And we use in Solexa, and of course, there is noise on this technology, but Adam has used a number of statistical approaches and to clean the data and to be able to extract some of the information at the population level in these viruses. And so here you have a coverage. One of the advantages of doing deep sequencing is that you can get an average of 100,000 coverage of the genome. So this you sequence the same position, between 50 to 500,000 each position in the genome. And so these are different populations, and you see it's very consistent what we've been getting. And so here's the first interesting result of this sequence. I don't know if you can see this, but these 3 lines here is the sequence, the SNPs, so the variants that we documented, the mutations that were accumulated with respect to the consensus of wild type, max, and SD. In other words, each of these lines correspond to one variable that appear in the population. And if the line is red, this value, we use a sort of analysis called Blossom 62, and what you see is as the more red it gets, the more different the amino acid that has been incorporated with respect to the original amino acid or nucleotide. So for example, in here, you have a lot of red, and whereas when you compare with SD, there is less red. So clearly, the patterns in SD and where the virus goes in terms of accumulating mutations is very different than what wild type can do. In other words, wild type can actually accumulate some mutations that SD cannot accumulate. In this particular analysis, we are comparing the capsid with the non-capsid, and you see many more overlapping mutations in the non-capsid region with respect to capsid. So again, it seems that when you mutate away from, or move away from the sequence space, now you're starting to have different type of mutation accumulating in this region. Finally, the robustness correlates with virulence. So in here, you see wild-type max and SD in mouse, and you see the SD is attenuated with respect to the other two. And you also see that it's almost significant difference with respect to the titles in brain. So I'm going to skip the conclusions, and I'm just going to say that this has been project that has been single-handled by Adam, Lauren, Ashley Acevedo has helped him for the mouse experiments. This was supported by NIH and UC research program, and we have this collaboration with Joe DeRisi for the sequence, micro array sequence. And thanks very much.
Also influences this. It would predict that when you look at wild-type isolates, they would be pretty central and pretty similar in what the genotype looks like. So, because you would go approach one in your diversity. Am I right? So
from a clinical point of view, is obviously understanding what can the virus do in terms of escaping, say, antibody or T-cell responses. Understanding what rules, what are the things that govern the ability of the virus to adapt, is critical. Now, can we predict evolutionary trajectories in a clinical setting? And probably, I know at this point, but I think as the sequencing technologies are improving, probably we'll be able to now starting to have a better sense of what the virus can do in terms of escaping one response or the other.
What about deletions or duplications of the genome? Have you looked at that?
Was that?
Deletions or duplications of the genome instead of deletions or duplications of the genome? Have you looked at that? Do they accept those type of mutations?
I'm not sure if, well, in general, the virus is very susceptible. If you have deletions or duplications in general, the virus will have problems because it's a single polyprotein, a very simple virus. So in general, you don't see this kind of alterations. And I just was wondering whether our analysis of the sequencing data will allow us to detect those. In general, if you have one nucleotide or two nucleotide deletion, the virus will not be viable because you have the whole polyprotein out of frame. But there are cases where you document that viruses have deletions in non-coding regions, and those have profound consequences for the biology of the virus, the replication of the virus.
A quick question.
Thanks.
Do we have time for another question? Over here. Yes. Yes. I'm just wondering, have you looked for any positive selection? I would expect that you may get some ribavirin-resistant mutations to show up.
Positive selection.
After applying the mutagen ribavirin, do you see positive selection of ribavirin-resistant viruses?
You mean if you get viruses that now replicate better in the presence of ribavirin?
Yes. Yes.
Yeah. We actually select. That was the base of the selection for viruses that have high fidelity. It was using resistant ribavirin as a way to force the virus to introduce less mutations per replication.